This will be the second and also the last video in photosynthesis, so that's the third part of Unit 3 of the National 5 Biology course. So this is going to focus mainly on measuring rates of photosynthesis and also the limiting factors of photosynthesis. So you need to know what a limiting factor is. So a limiting factor is basically anything that is in short supply that is needed for a process. Okay, so if you think about photosynthesis, it needs carbon dioxide, it needs water, it needs enzymes, it needs light, it needs chlorophyll, etc., etc. Okay, now there are three things within that that are essential, that without those, or if those are in short supply or those are limited, photosynthesis will either slow down or it'll entirely stop. Okay, now these three limiting factors are really important. So the three ones are, and wording of this is key, okay? Carbon dioxide or CO2 concentration. Don't just say CO2, that's not enough, okay? So concentration is essential, because it's how much of that CO2 it's getting, okay? The next one is light intensity, not just light, but light intensity, how strong that light is, how much light they're getting. Okay, and the last one is temperature. If it's too cold or it's too hot, think about the relationship with that and enzymes, okay? Because that affects enzymes. Right, now the reason why this is important is because if these are in short supply, photosynthesis is slowed. And it's not just then photosynthesis that slows, it's growth that slows as well. So then plants can't grow correctly and grow sufficiently, and then ultimately might die. Right? So this is important for us to know just in general in terms of production of food, but it's important to relate it back to what we talked about in terms of the different factors involved. Now, if you think about rate of photosynthesis, you can measure it in a few ways. Now you can measure it in the things that it takes in or the things that it gives out, okay? So if we think about the things that it takes in, we can measure photosynthesis by measuring the CO2 intake in that plant. Okay, so basically if we change the concentration of CO2, that then can measure how much CO2 is taken in, or we can adjust it and then measure that. Okay, that's not a common one, so it's not one that you'll tend to see very often. The next one is about measuring things that it gives out. Okay, so the oxygen production by a plant is a really easy measure of photosynthesis. Now either that can be volume or it can be number of bubbles produced per minute, and I'll explain what that means in a sec. And the last one is the carbohydrate production. Now that basically means how much is produced after it's photosynthesized. So you wouldn't really tend to measure the sugar involved in that because sugar tends to be stored or used in something else. So what you can do is measure the dry mass of a plant. Okay, and that will then determine whether it increases. It means it's storing more starch, for example, or it's making more cellulose and therefore it's photosynthesizing. Or if it's lowering, then it's producing less of that. Now the reason why it's dry mass is because water levels in a plant will change over the day. Okay, so if you decide that you're going to use wet mass, okay, without drying that plant, then that water level will change across the day, which is not really useful for us. We can't then we can't determine whether we're measuring water or whether we're measuring the actual mass of that plant. Okay, so all these can be used to determine the rate of photosynthesis, how fast it's going. Right? A plant that is not photosynthesizing very fast won't take in much carbon dioxide, and it won't produce much oxygen, and it won't produce much carbohydrate. Okay, so what I'm going to do now is show you a quick setup of an experiment, which is often shown to show the rate of photosynthesis. Right, now this is a beaker of water. Okay, this is an aquatic plant. Now, there's plenty of different species they use. Sometimes they use one called kabomba. There's loads and loads of species. So in the example, if they give you one in the exam, then that would be, they would just name it and it wouldn't really matter. Okay, this is a syringe. And that would be used to collect the gas. Or we could be using measuring gas in a different way. And this is a lamp, which I'll come on to in a wee minute. Now, with this, just as an example, okay, if we were looking just to measure the rate of photosynthesis in general, that plant would sit there, okay, and it would eventually produce oxygen, and that oxygen would bubble out of the leaves, and we could count it, okay, count those bubbles being produced over a minute. Okay, what you could also do, rather than counting the bubbles, because that's quite difficult, bubbles are different sizes, they contain different volumes of gas, right, is we can leave that oxygen to bubble up and then fill a syringe. And that could be used to measure the rate of photosynthesis, because that would change, it would move from here, for example, to here over a period of time, and that would measure your rate of photosynthesis. Okay, so how much oxygen it produces gives you an indication of how fast it is photosynthesizing. Now, if we have a lamp here, we can use then and change this position of this lamp to determine whether light intensity affects photosynthesis. So if we have a lamp here, it'll be giving it a lot of light energy, okay, and a high light intensity, and this plant should photosynthesize quite a lot and produce a lot of oxygen. When we move that lamp away, okay, and put it along here, just as a quick example, right, if that lamp is moved further away, this would be low light intensity, and this would be high intensity. Okay, so as we move that light away, the plant will produce less oxygen, and when we move it closer, it'll produce more. Now, there might be an issue with this setup, okay? So think about the fact that lamps often produce heat as well as light, and you should think to control that, okay? So there should be really some kind of heat shield placed in front of this. Okay, and that prevents the effect of temperature, and therefore make sure that you can have valid conclusions, valid comparisons between your results. Because you then, if you don't have that heat shield, can't tell whether it's the temperature or the light that is causing that change in the rate of photosynthesis. Okay, now think about the other factors that you have to keep the same. It would have to be the same species of plant, same rough size or mass, or number of leaves, or the surface area of the leaves. You'd have to have the same length of time which the experiment runs for. Okay, there's lots of things that you'd have to keep the same. The lamp would have to be kept the same, okay, but the distance would be changed, for example. So this experiment, you can change a lot of things about it to investigate different factors, but this is just a very general example. All right, so now that I've shown you a quick setup of it, what I'm going to do is talk to you about the limiting factor graphs. So limiting factor graphs are quite common as multiple choice questions. Now, for this, you actually don't really have to understand a lot about them, because a lot of the time it is just pulling data from what they've given you. So graphs can be shown as just one factor on a graph, or they can be shown as multiple. So I'm going to go through quite a number of examples. In this very first one, we have temperature on the x-axis. Okay, so I'm just going to put some paper here to get rid of this stuff now. This temperature here is too low to have photosynthesis at its maximum rate. So here at point X, it is temperature that is limiting the rate of photosynthesis. And here, as temperature is getting too high, it is also limiting it at this point, because it's starting to denature the enzymes. All right, now this is probably the most simple graph that you're ever going to come across, because if it looks like this, It's temperature that's limiting at every point. Now the next example I've got here is one that shows carbon dioxide concentration and rate of photosynthesis on the y-axis. So here, rate of photosynthesis is increasing because the more carbon dioxide you give it, the faster it can go. So anytime that you have got a point on the slope, it is whatever's on the bottom of the graph, whatever's on the x-axis, that is limiting it. So CO2 concentration is limiting it here. Now at this point, photosynthesis rate has levelled off. Now it's levelled off because it has enough carbon dioxide, but it's been limited by one of the other factors. So our three limiting factors are carbon dioxide concentration, temperature, and light intensity. Okay, so we've used carbon dioxide concentration here already, and it has enough carbon dioxide to be photosynthesizing at its maximum rate. So therefore Y could either be temperature, or it could be light intensity. Now you don't have enough information to tell them whether it is one or the other. So you just say either or, okay, or both potentially. Again, another example here shows light intensity. So what I said earlier on, whatever's on the slope of the graph is whatever's along the bottom of the graph, okay? So X is light intensity because at this point it's increasing light intensity and that rate of photosynthesis is getting faster. So that's the thing that's holding it back. Now, once it levels off and that rate of photosynthesis stays the same, that means that light intensity is no longer limiting it. Something else is limiting it. And that something else is either one of the other two factors that's left over. So if we have light intensity already been used here, the other two factors left over are CO2 concentration, or we have one more, which is temperature. Okay, and as a quick final example, I've got one more down here. Now that looks slightly different to what we had, okay? And it's because there's three letters that we're looking for you to do. Now again, same as applies before. X, whatever's on the slope, is whatever's on that bottom of the graph, okay? Because this is at low concentrations and photosynthesis is low. This is at high concentrations and therefore photosynthesis is high. So this is CO2 concentration. Now again, the same as we've got up here, Y and Z, we don't know, they've not given us any other information. So Y and Z could be either, doesn't matter which, light intensity, which is one of your other limiting factors, or it could be temperature. We don't know. So the answer for either of those, Y could either be light intensity or temperature, and Z would be whatever one you didn't use for Y. Okay, so if you said Y was light intensity, Z would be temperature. Okay, or vice versa. It doesn't matter which one you put down which way. Okay. Now, these are actually really straightforward when it comes to answering them, especially if they give you a bit more information. So the next example I'm going to show you is with two factors on the graph. Okay, and again, it's all about reading the graphs. It's not even about understanding it. So two factors on the graph. Right here we've got carbon dioxide concentration, and here we have lux, which is light intensity. So whatever's on the slope of the graph, which is X, is whatever's on the bottom of the graph, whatever's on the x-axis. So this is CO2 concentration. Now for this graph, they've given us more information. They've given us light intensity. So if light intensity is low, this is the rate of photosynthesis, but when they increase it, that rate of photosynthesis increases. So that's showing that this light intensity is holding it back here. So this is light intensity. Okay, now this is just reading the graph. Whatever's on the slope is in the bottom of it. Okay, whatever's on the flat is whatever's on the side. Same idea here. Whatever's on the slope is whatever's limiting it at the bottom. Okay, so light intensity is limiting it at X. So this is light intensity. Y is whatever's on the side. So it's temperature. Okay. Right, they've not given you any information about light intensity, so you can't use that, any information, sorry, about carbon dioxide concentration, so you can't use that as an example. Right, the last one, X, whatever's on the slope, is whatever's on the bottom of the graph, so X is CO2 concentration. y is whatever's on the side. And again, whatever's on the side is temperature. Okay, so these are actually more easy than the ones before because it gives you more information, so you don't have to really think about it, you just have to read off the graphs. Now the last examples look a bit more scary, but again, they're just reading off the graphs. So this one shows all three limiting factors on a graph. Okay, we have light intensity along the bottom, and we have temperature up here, and also carbon dioxide concentration. So same procedure as before. Whatever's on the slope of the graph is on the bottom, okay? So X is light intensity. Right? Now, what you're looking for at Y is whatever has changed between Y and then moving up to this next stage. So when we want to move from Y up to Z, okay, that rate increases. Now, why does it increase? What has changed between Y and moving up the way? The only thing that's changed is temperature. Okay, carbon dioxide concentration hasn't changed, so that's not caused the rise. So Y is temperature. Okay, to get Z to move up, you look at what's changed. Temperature hasn't changed. Carbon dioxide concentration has changed. So Z is CO2 concentration. Okay, so you're just looking at the details they've given you in the graph. So as another example here, in the last one that we're going to do, X is again, whatever's on the slope is on the bottom of the graph. So X is CO2 concentration. Now if we look at Y and we look to try and move it up to the next level, what has changed to move it up? Y is temperature, because it's changed from 12 degrees to 14 degrees. Light intensity hasn't changed, so Y is temperature. Now again, same logic. To try and get Z to move up, what would you then change? What's changed? The lux, the light intensity has changed. Okay, now these graphs, like I said, it doesn't require understanding, it just requires reading and interpreting the graphs. Okay, these ones can be quite tricky just because there's quite a lot of information. Take your time and read it carefully. Okay, because there are quite common questions in exams and homeworks and things like that. Okay, and that finishes off the limiting factors video for photosynthesis.